Hello, everyone. Hope you are all healthy, not coughing too much. It's Monday, November 16th, 11 o'clock. Now tell me if you can hear me.
Yes.
Thank you. Good. So we finished chapter 10 and chapter 11 will be the subject for this week. I don't know if we're going to finish it on Wednesday. We may actually take some time from next Monday as well to finish it. Now the homework is open for chapter 11. So you can start working on it right after this lecture if you want. So it will be available for a week. Oh, wait, Wednesday. No, Wednesday. Next Wednesday is we don't have a class next week Wednesday, right? Am I correct? So let's see how many classes we have left. Right, so we have this week 16, which is today, and 18, Wednesday. And then 2 more lectures on bias signaling. One is November 23rd, which is the first day of Thanksgiving week. Then there's a break, and we'll have a lecture, the last lecture on Monday after Thanksgiving. So we don't have that much time, so I have to be quite selective in terms of what we cover. And let's get going. There was a paper I wanted to discuss with you, which is a very interesting paper just to show you how some recent research is done in the area of lipids, which is very pertinent, very pertinent to our class. Can you see this paper on your screen? Hello?
Yes.
Thank you. So this paper is in journal. We haven't done this yet, right? Just to make sure I'm not getting anything confused. You haven't seen this paper yet, right?
No.
Okay, good. So I uploaded this to Canvas. And so what this deals with is the detection of sphingosine in living cells. Remember I told you that the metabolism of lipids defects in this metabolism can lead to a lot of various health issues, right? And so there are ways to diagnose that and predict the outcome and severity of various diseases by measuring the levels of lipids in cells, lipids that have not been metabolized, for example. So sphingosine by itself. So let's see, abstract. Now, first of all, the paper is in JACS, Journal of the American Chemical Society. It's a top journal in chemistry. Obviously, Nature and Science are much better. These are 2 top journals, but as far as chemistry, this is considered this, and there's a German journal, Angewandte Chemie, 2 top journals in chemistry in terms of impact factor. So, so this is a very recent paper. It came out just, I don't think it's the recent issue. I think it's the issue right before the recent one. I was just looking through some papers and found this one. So, so sphingosine, we know it's a structural lipid, right? And it's also a signaling molecule. And abnormal sphingosine metabolism is observed in several diseases: cancer, diabetes, Alzheimer. So, however, despite of this importance, there is a lack of tools for detecting sphingosine in living cells. And in general, there is a broader challenge of developing highly selective live cell compatible affinity probes for hydrophobic lipid species. Why do you think it's harder to detect hydrophobic lipid species rather than, I don't know, carbohydrates, for example, or protein, for example? What is it about lipids that makes them hard to detect? Yes, just think about intramolecular interactions. So usually you detect lipids with you detect species with something that can make bonds with it. No? Nobody? All right. So lipids don't make intramolecular bonds very easily, right? If proteins, for example, you can have a probe for it, a small molecule that can bind protein through hydrogen bonding or through electrostatic interactions, carbohydrate, hydrogen bonding. Lipids don't really form strong bonds, right? Because it's primarily hydrocarbons. And so in general, there is a lack of such probes. And that's why this paper is important. And so, so these scientists now, Neil Devaraj is at UCSD, as far as I remember. Yes, Department of Chemistry, Biochemistry, University of California, San Diego. So they developed such a small probe and detected it and used it to detect endogenous pools of sphingosine. So they successfully detected, for example, sphingosine accumulation in cells from patients with this particular type, disease type, Niemann-Pick type C1. It's a lipid disorder in which increased sphingosine mediates disease progression. All right, so let's look at how they did it. So the probe was actually this particular salicylic aldehyde. You can see here there's a benzaldehyde group, right? And there's a Q and F. So F is a fluorescent dye shown here, okay? And Q is a quencher. So it's a molecule which will quench the fluorescence. So you can see here the idea is that sphingosine, shown here now as SPA, it's a minor variation of sphingosine that has no double bond, but sphingosine is much more prevalent. There's about 10:1 ratio of sphingosine to SPA. And so they took advantage, like you see, there's an amino group and there's a hydroxyl group. An amino and a hydroxyl group will react with the aldehyde to form this mixed acetal where you have, remember in acetal, or you will have two oxygens. So here there's one oxygen, one nitrogen. So it's a mixed acetal with oxygen, nitrogen participating. So once that is formed, you can see that this nitrogen is in a very close proximity to the ester carbonyl. It will attack the ester carbonyl, form an amide bond. This mixed acetal will hydrolyze, and sphingosine will basically leave with the fluorescent molecule attached to it. But net result is that we separated F and Q. You see Q and F were part of the same molecule, and so the quencher was quenching F, fluorescence, because it was in the same molecule. Now sphingosine came in, stole this fluorescent moiety, ran away with it, and the fluorescent moiety started to fluoresce. There's no more quencher. And so if you look here, the aldehyde group is crucial. So usually for experiments like that, you use controls. And you can see here the R is the aldehyde in compound 1, and it's a hydrogen in compound 2. So in other words, this probe should work with compound 1, but should not work when the R is H in compound 2. And we can see it here. So this is a relative fluorescence depending on the wavelength. You can see in control, we see very little fluorescence. Control is compound 2. And compound 1, we're going to be increasing. You can see at 0.25 millimoles sphingosine, we have the highest fluorescence response. Highest fluorescence response. You can see this in cells. So these are HeLa cells, right? HeLa, you know that it's a cervical cancer cell line. And we're going to be increasing the amount of sphingosine in these cells. You can see in the probe, you can see nice fluorescence as you increase the dose of sphingosine. But in control experiment where there's no aldehyde, there's no increase in fluorescence. You can see there's also quantitative representation. So these bars represent fluorescence, right? So the taller the bars are, the higher the relative fluorescence. And so this is with 1, and this is with 2. And 2 is the one that has no aldehyde group. So there is no relative increase in fluorescence. And so let's go straight to, so now these are endogenous HeLa cells, right? Same idea, compound 2 and compound 1, high fluorescence, compound 2, no fluorescence. And you can see here by this bar. And finally, they isolated fibroblasts. They isolated fibroblasts from patients. These fibroblasts, let's see. Yes, we cultured fibroblasts derived from healthy and patients who were afflicted with this disease. So in other words, they collected cells, fibroblast cells, which are afflicted with this disease, which have a defect in metabolizing sphingosine. And you can see here, so these are healthy fibroblasts, and these are NPC1 fibroblasts. So you can see again, large difference in fluorescence response, right? So these are healthy, and these are diseased cells. So what do we learn here? Now let's look at some of the conclusions. The probe is sufficient to detect biologically relevant changes in sphingosine levels, but it has limitations, right? So it has not made it into the clinic yet. It's just a paper in the Journal of American Chemical Society. So what stands in the way for this molecule to make it to human clinic? First, it's subject to hydrolysis under biological conditions. So if we look at the structure, here, which group do you think will undergo hydrolysis? So compound 1. Which one is hydrolytically unstable? Yes, anybody? You guys are quiet today.
The ester that's next to the fluorescent molecule?
Yes, we know that esters are hydrolytically unstable. So this ester will undergo hydrolysis, and what will that do? So if the ester undergoes hydrolysis, what will happen to fluorescence?
It would steal the fluorescent molecule before the lipid could, is that right?
Yeah, so the fluorescent molecule will depart, will leave. So the quencher and the fluorescent molecule will separate. And what will happen in terms of fluorescence of the mixture of the cells?
I'm not sure. Would it fluoresce if if it leaves, or does it only fluoresce if it's attached to the lipid?
No, it's a dye. It's a fluorescent dye. It'll fluoresce.
Okay.
Yeah, it will fluoresce by itself because the quencher is no longer there, and so there will be increased fluorescence in the absence of sphingosine, which is what's known as increase in background fluorescence, right? So background response is usually the response which occurs in the absence of the analyte. If the analyte is absent, but you still have a lot of fluorescence, which comes from this hydrolysis, then obviously that limits, that reduces the detection limit of your probe. All right, the reaction rate of our probe is sluggish and requires long incubation times. That's also an issue, right? So we don't want the patient to wait for days to find out his levels of sphingosine if the treatment is required quickly. Third, quenching of BADIPI. Now, this fluorescent dye is known as BADIPI. I did not actually give you the name of it, but it's called BADIPI. Okay, the quenching is not complete, right? So again, maybe a better fluorophore quencher pair can be developed. And finally, low dynamic range, resulting in a small difference in fluorescence intensity between normal cells and cells that with gross changes in sphingosine levels. Now, if you look at this graph here, now, yes, you can look at this and you say, oh, this bar is so much lower than that bar, then the fluorescent response must be high. But that's not true. If you look at the relative units, what is this? This one is 1.05, and this is 1.18 or something like that, right? So they're very close. It's not like this is 1 and this is 10, right? So often don't get deceived by this kind of presentation, right? When the units are so close, even though in the picture it looks beautiful, but if you look at the units, they're so close, which means that the compound becomes fluorescent upon reaction with sphingosine, but the increase in fluorescence is not as high as you wanted it to be. All right, do you have guys any questions about this paper? I think it's a very nice paper, highly relevant to what we're discussing. And if you don't have any questions, let's move on with the material. Obviously, I may ask some questions about this paper on the test, so hope you understood that material. If not, let me know. All right, so chapter 11, what are we gonna learn? All right, we're gonna learn so we're gonna be talking about biological membranes and specifically transport across the biological membranes. Structure and composition, what kind of lipids they consist of, what kind of proteins are embedded in biological membranes. We'll talk about dynamics of membranes. So these lipids, they're not static, they move. And we'll learn about where they move and how fast they move, right? So biological membranes are not just frozen species that have no movement inside. It's important to realize. Now, structure and function of membrane proteins. So what membranes are inserted into biological membranes and transport. All right, so there are three major structures as far as aggregation of lipids, right? So we do know that lipids will aggregate in water, right? They have a hydrophobic portion and hydrophilic portion. And so there are three major types which are observed. Micelles, we already talked about micelles a little bit. bilayers, which are biological membranes. And there's another one we haven't talked about, liposomes. Liposomes are, it's a new area of research, important in, for example, development of drug delivery methods. We're going to look at that as well. And obviously, as far as which structure is formed, that depends on the type of lipid and also depends on its concentration. Okay, micelles. So micelles are amphipathic molecules that have a larger polar head group than tail, right? So the polar head group is big, then it forms some kind of a, I guess, a wedged shape structure, which is very favorable for the formation of this kind of spherical aggregate. Okay, so the head group is bigger than the side chain, and so fatty acids will form like that, right? So we talked about soap, and that's what soap makes in water, right? So you can see the polar head groups of soap will stay on the outside, interacting with molecules of water, and all sorts of hydrophobic stuff, such as dirt, when you wash your soap, will go inside. Or various infectious particles, for example, can go inside your COVID, right? Can go inside. So, and the aggregation is concentration dependent. So you just need there's a what's known as a critical micelle concentration, CMC. If the concentration is too small, then there's not enough for the fatty acids to aggregate. They would rather just float by themselves in water. But once you reach the critical micellar concentration, then they will form micelles in water. All right, so the second one is membrane bilayer. So here the individual units are cylindrical, right? So the crosshead, the head group in general should be similar in size to the side chain. And you can see here there are two fatty acids. There are two fatty acids per one head group. In micelles, we only had one. Well, I don't know why they're shown. Maybe there are. Anyway, in general, in micelle, there should be one fatty acid, right? We'll talk about such lipids later. But so that what makes these lipids form bilayers because of the similar sizes of the hydrocarbon tail with the head group. Now, the two types that we're gonna discuss in great detail are phospholipids and sphingolipids. And specifically how we already know about them obviously, but how they participate in the function of membrane bilayers. Okay, so hydrophilic head groups interact with water on both sides of the bilayer, and hydrophobic fatty acids are packed inside. So this was the third one, liposomes. So these form vesicles, right? So here the again, the difference between this and the micelle is that there is an inner cavity shown here, right? So you have a lipid bilayer as opposed to monolayer in micelle, lipid bilayer, and head groups inside of that, and there is an aqueous cavity which can actually store materials inside. For example, you can use this and transport a drug. So artificial carriers. And so the way actually they will bring the drug is because they can fuse with the cell membrane. They can fuse with the cell membrane and release the cargo. So, membranes. Lipid-based structures that form pliable sheets, composed of a variety of lipids, lots of different proteins embedded in them. So we know that all cells will have a cell membrane, which will separate the cell from its surrounding. And eukaryotic cells have various internal membranes, right? So there are inside the cells, there are lots of organelles that will divide this space into these compartments. You can see here electron micrograph of biological membranes. So for example, here, these wavy lines is membranes of endoplasmic reticulum. Now there's a membrane associated with the nucleus, a mitochondria. All these are separated by membranes. There is secretory granule, right? So there's stuff inside them which has been produced and can be released to the outside extracellular environment. So lots of membrane, not just on the outside, but lots of membranes inside the cell. So what do? Well, they define the boundaries of the cell, right? So they define a space that will be occupied by the cell. They allow for import and export, right? So nutrients will come in, waste and toxins will leave. They will retain molecules and ions within the cell. They will not let them out, right? Metabolites that are required for the cell, various ions which are required for the functioning of the cell. They'll be retained inside the cell because of the membrane. They sense external signals and transmit information into the cell, right? So they provide a way how a cell communicates with the outside world. Provide compartmentalization within the cell. And you can see here, for example, energy producing reactions from energy consuming ones, right? So you don't want the two types of reactions to be in the same organelle, to be in the same space in a cell, right? So these have to be separated, otherwise they will kill each other, right? If this produces energy, then immediately there's another reaction that will consume that energy, and the result will be nil, right? Now, for example, also proteolytic enzymes, such as, for example, enzymes in lysosomes, right? That whose function is to recycle, let's say, proteins, to chop them up into pieces. We don't want these proteolytic enzymes to chop up important cellular proteins. That's why they're kept in lysosomes. Produce and transmit nerve signals. So these membranes are important for ion mediated action potential in nervous system. Store energy as proton gradient, support synthesis of ATP, lots of different functions of membranes. So some common features. So they're about 30 to 100 angstroms thick. 2 leaflets, so we just looked at that. And we will also see that archaea, now archaeobacteria, that's an old term, because actually archaea is now considered these are prokaryotic organisms, but there are differences between them and bacteria. So oftentimes people just refer to them as archaea now. not archaeobacteria. So these form a monolayer of bifunctional lipids. Okay, so in other words, these will have two head groups and one hydrocarbon piece, right? So these will be monolayers. So we will look at this the picture of one of those. So form spontaneously, hydrophobic effect, we know that. Protein molecules span the lipid bilayer. Now it's important to realize that membranes are asymmetric. We're going to look at that. What that means is that there's an inner leaflet, there's an outer leaflet, and the type of lipids which are inside or outside are different, right? They're not just randomly located in both leaflets, right? So there's some lipids are found on the outside only. Some lipids are found on the inside only. And that's very important for cell physiology. And we'll look at that as well. And the fluid structure. So basically, two-dimensional solution of oriented lipids. They're not frozen. All right, so there is a fluid mosaic model of membranes, which is what is currently accepted as the description for cell membrane. They form viscous two-dimensional solvent into which proteins are inserted or integrated more or less deeply. Okay. So there are integral proteins which are firmly associated with the membrane, often spanning the bilayer, and there are peripheral proteins which are weakly associated and can be removed more easily. So proteins covalent, non-covalently attached, and there are proteins which are linked to membrane lipids. So let's look at some representations of those. So this is the fluid mosaic model. So you can see now these are lipids. Now there are two kinds of lipids, as we talked about. So the blue ones will be glycerophospholipids, or phosphoroglycer, no, what do you call that? Glycerophospholipids, I think that's how it goes. So the blue ones, and there are sphingolipids, which are the red ones, right? So they both are main components of the bilayer. Now, what else do we have? We have a glycolipid. So for example, this lipid is attached to an oligosaccharide. We have these banana-looking things, which are sterols. So we looked at some structures of those in the previous lecture, in the previous chapter. So this is an integral protein, which crosses the membrane, and it crosses the membrane in this alpha helical fashion, right? So obviously you might already consider that the amino acids that span the cell membrane, are they hydrophilic or hydrophobic? What kind of amino acids can you find in this alpha helix? Just give me examples of 3 amino acids that you will find in this alpha helix. Need to wake you up. We've been quiet today. So.
What, phenylalanine?
Maybe. I don't like. Yeah. Phenylalanine is good. A good one. And the reason why you chose it?
Because it's non-polar, so it'll be hydrophobic.
Yeah. Give me a couple others.
Glycine or proline?
Yeah, proline is good. Well, proline. Now think about proline and alpha helix.
It was the one that would make the turns, so maybe not proline.
Yes, yes.
Okay, got it. So maybe leucine instead.
Leucine is good, yeah. And probably not glycine either. Glycine is not very hydrophobic. Remember, it doesn't have a non-polar side chain. And also it allows for a lot of flexibility, which alpha helix doesn't like. It likes interlocking between amino acids, right? So leucine, and the third one?
Arginine.
Not arginine. Where will you find arginine? In this protein, where will you find arginine?
It would be outside of the cell or outside of the membrane.
Mm-hmm. Could be here or there. But so not arginine there. How about something like isoleucine? Will that work? Or valine? Yeah, so those are hydrophobic amino acids. All right, so sterols, then this is a funny chain-linked protein attached to a lipid covalently. This is a peripheral protein, which is just attached to the polar head groups, right? Doesn't really go into the membrane easily. Now, this is peripheral protein covalently linked to a lipid. It has a tail, hydrophobic tail, that it inserts into the bilayer, and this tail holds it in place, right? Because it interacts with the other lipids inside the bilayer. All right, and then GPI anchored proteins. So we talked about those GPI anchored proteins. Maybe we'll talk about them a little more. All right, any questions? What do you think that thing is?
Like a channel?
Yeah, that's what I think. It's not labeled, but I think it looks like a channel to me, right? So it's like an ion channel, like an ions can actually slide in and come out on the other side, right? This is a funny one. I have no idea what this is. Looks like it's a channel with something in it. No? Some kind of molecule in it. Maybe it's a transporter. Maybe it's a protein transporter that has, for example, glucose that it allows for its passage. All right, so composition of membranes. So lipid composition varies by organisms, tissues, organelles. Ratio of lipid to protein changes, right? So depending on the organism, tissue, organelle, everything changes. Cholesterol predominant in the plasma membrane, virtually absent in mitochondria. Galactolipids, for example, are in plants, but almost absent in animals. So you can look at all those. Now, membrane composition variable in different organisms. So for example, human myelin sheath. Now remember, the function of this is to protect or insulate nerve cells, right? And so you can see actually, based on that, there is not so much protein compared with the other, right? Not so much protein, mostly phospholipid and sterol, cholesterol. So which is used for insulation. Now, more functional membranes, more functional membranes, for example, such as yeast, that is a unicellular organism that requires a lot of communication with the outside world, right? It has lots of proteins and very little phospholipid. And remember, it has ergosterol as opposed to cholesterol. different type of sterol. And different organelles as well. So you can see actually the two most abundant phospholipids are this light blue, which is phosphatidylcholine, and the dark blue, which is phosphatidylethanolamine, right? So PC and PE. These are two most abundant ones across almost all organelles. Cholesterol, there's lots of cholesterol in the plasma, but very little, for example, in mitochondria, tiny bit. But there's lots of cardiolipin in mitochondria and almost none of it in plasma membrane. So anyway, that's just nothing really for us to memorize here. Just remember that phosphatidylcholine and ethanolamine are most abundant. That will be good enough. Membrane composition and asymmetry. So you can see here that so this is a nuclear envelope, which is connected to endoplasmic reticulum, right? So endoplasmic reticulum has all these different kinds of lipids. So the lipids are shown here. So the one with the OH is going to be the ceramide. The ceramide there is some cholesterol in the ER, and there are different kinds. I can't really tell the color, but different kinds of lipids. So so these kind of lipids then can bud off the endoplasmic reticulum. And as you remember, many proteins which have been folded in the endoplasmic reticulum then move on to Golgi apparatus, where they often undergo post-translational modifications. And you can see here, basically these vesicles kind of travel within the Golgi apparatus. And eventually, after the complete protein has been formed, they're packaged in secretory vesicles and are allowed to leave the cell membrane. So, for example, this kind of lipids these kind of lipids are representative of Golgi and plasma membrane. There's a lot of these, the red ones, which are sphingolipids. And there's something you can see here. Many sphingolipids have longer fatty acids, right? So if you look at the glycerol phospholipids, their fatty acids are a little shorter, and sphingolipids fatty acids are a little longer, and that actually results in thickening of cell membrane in this area, right? And this is what's known as lipid rafts. And we're going to talk about lipid rafts on Wednesday, I think. Rafts. Okay, so membrane bilayers are asymmetric, I told you, right? Two leaflets have different lipid compositions. So the outer leaflet is often more positively charged. Now, phosphatidylserine outside has a special meaning. So the platelets activates blood clotting. Other cells, it marks cells for self-destruction. So, so phosphatidylserine primarily resides in the inner leaflet, but if a cell undergoes apoptosis, for example, if it needs to be engulfed or eaten up by phagocytes in the body, then phosphatidylserine will undergo a flip, right? Will undergo a flip. We'll talk about the enzymes which accomplish that. They're known as flipases. They will move phosphatidylserine to the outer leaflet of cell membrane, and those will be like flags. Like flags, hey, I am dying, I want to die. And different phagocytes, like macrophages, for example, they will come in and engulf these cells. So that's why apoptosis is commonly referred to as a clean cell death. So normally, if something dies in a non-programmed way, that leaves a lot of cell debris. And then that causes inflammation. So that's not good for the organism. But apoptosis is a nice, clean way to die. It's sort of the cell is already taking care of its own debris, right? It allows for the phagocytes to come in and clean up the mess. So archaea, multiple membrane constituents. So phospholipids, L-glycerol in archaea, D-glycerol in bacteria. So remember that glycerol is has a chiral center. So let's say it's a phospholipid, right? R1. R2. And there's a hydrogen here. There's a hydrogen, so this one is a stereocenter. Chiral center. And so, so in archaea, so this is primarily in an L configuration. In bacteria, for example, it's in a D configuration. Branched isoprene chains in archaea, unbranched fatty acid chains in bacteria. Ether linkages in archaea, ester linkages in bacteria. So we talked about ether lipids a little bit. And also membrane topology, monolayer in some archaea. And I think we have a picture here. So this archaea lives near volcanic hot springs, high temperatures, acidic environment. And you can see what's happening here. So there's a hydrocarbon segment here, right? And then two glycerols, one on this side, one on this side, and there's actually a third one which is not participating in the connecting itself to hydrocarbons. But you can see they're branched. And so now tell me if this is an ester phospholipid or is this an ether phospholipid?
Ether phospholipid?
Ether. And why do you think this would be ether? Like, what does archaea gain by utilizing this ether phospholipid?
It's more stable, like less subject to hydrolysis than an ester linkage would be.
Yeah, and why is that needed? Why do archaea, like this particular one, why do they need these lipids to be more stable?
Because of that harsh environment that they're in, since it's more acidic,
Yep.
Is that it?
Yep, I think so. So high temperatures, acidic environment, so ether lipids are much more stable. And so the cell membranes are much more stable and the cells are much better protected from the harsh environment. Yeah. Sounds good. All right. Proteins. What do proteins do in membranes? Detect signals from the outside. We talked about light when we talked about lipids, right? We talked about light and how opsin combines with rhodopsin to perceive light. Now, hormones, for example, insulin receptor is in cell membrane, right? And it's waiting for insulin to arrive and tell it to start the process to intake glucose from the bloodstream. Neurotransmitter, acetylcholine receptor, right? So how the nerve signal transduction occurs. And so obviously the receptor for that will be in the postsynaptic cell, right? Presynaptic cell will release acetylcholine, and postsynaptic cell will have the receptor for it. And it will propagate the action potential. Pheromones for taste and smell, different channels, gates, pumps, important enzymes. So we'll talk, these kind of enzymes we'll talk in biochemistry too, if you take that. They're important enzymes for a variety of processes. For example, lipid biosynthesis or synthesis of ATP in mitochondria. All right, so there are various types of membrane proteins. So we already kind of mentioned that there is an integral. There are integral proteins. Now, some of them span both layers, and so those are known as polytopic. Some of them are inserted kind of only into the one layer of the lipid bilayer. So those are known as monotopic. So you can extract those from the cell membranes using detergents. And these detergents, which have hydrocarbon tails, will interact with the hydrocarbon portion, or will interact with the nonpolar portion of these proteins, right? So this is nonpolar yellow one, this is nonpolar yellow one, and that's where the detergent will bind to these nonpolar and basically compete with the cell membrane for binding towards these transmembrane integral proteins. Now there is also proteins which are, you can see here, which are attached through the link to other proteins to cell membrane. And these are primarily through hydrogen bonding, electrostatic interactions, and you can break this up by changing the pH, for example. Chelating agents, urea. Remember, urea will break hydrogen bonds. Carbonate will do that also. Okay, so and second one is amphitropic proteins, right? So these proteins will, so for example, GPI anchor protein, so glycosyl phosphatidylinositol anchor proteins. So we talked about those. They have lipids and they have carbohydrates. And the protein is usually on the outside of the cell membrane. And you can see here there's an enzyme, phospholipase C, that can cleave this off and release the protein. And many proteins are associated with cell membrane, also known as amphitropic proteins. They bind to lipids, polar group, for example, phosphoinositol phosphate. Yes, phosphoinositol phosphate. So various ways of biological regulation can actually make this protein stick to it or come off. So peripheral, loosely associated, change in pH. And so that's what we just mentioned. Amphitropic proteins, conditional attached by interaction with lipids, carbohydrates to lipids. And can you see here, biological regulation can also play a role in making proteins attach or detach. And so here is the structure of the GPI anchor I was telling you about, right? So you can see here, it's a very common way for proteins to be attached to the cell membrane. So it stands for glycosyl phosphatidylinositol anchor. So here we have a glycerol moiety, 2 fatty acids, which will anchor the whole thing to the membrane. Then there's a phosphate inositol and a few sugars. It's an oligosaccharide, has a glucose and N-acetylglucosamine, a couple of mannoses, and then there is this another phosphate. Basically, this is ethanolamine, which forms a amide bond with the carboxyl portion of the protein. Okay, so this is probably the most complex way of protein attachment to the cell membrane. But proteins can be also attached through single fatty acid esters, right? So a palmitoyl group on terminal cysteine or serine. So you can see here, this is a thioester. Remember, this is an ester. And this one is a thioester. And so thioesters is another way for proteins to be attached to the cell membrane. And that will be accomplished through cysteine. Now this is a myristoyl group on the amino terminal, glycine, right? So these proteins which are attached this way, they have a glycine on their N-terminus, and they will be attached, they will make a bond with the myristoyl acyl group, a myristic acid, some cholesterol here and there, and farnesyl. Farnesyl, so it's a group on carboxyl terminal cysteine. Inside, outside. So some membrane proteins are lipoproteins, so that's what we just talked about. They contain covalently linked lipid molecules, long chain fatty acid, isoprenoids, sterols, and glycosylated phosphatidylinositol.
Quick question.
Yeah.
So what determines whether they'll be inside or outside of the cell?
Depending on what function they have. Depending on what function they have. So proteins in a lot of them are involved in recognition, right? A lot of them have to perform jobs at the cell membrane. So we will see, for example, enzymes such as ATPase, which is involved in energy. The such enzymes as flipases, which are important for lipid distribution in cell membrane, so cell recognition processes. Many of these proteins interact with the extracellular matrix, right? So they have to be on the outside because of that. Many of them are inside because, let's say, a signal arrives. For example, a signal arrives from this is outside, right? A signal arrives from the outside, and it talks to this. Let's say this one has some kind of hydroxyl group that can be phosphorylated, right? Then a different protein will come in, and the signal will be propagated. This protein will recognize the phosphate, and the signal will be propagated inside the cell. So it depends on what they do. Depends on what they do. So so farnesylation of proteins. Now here's, wait. For some reason, I don't have a slide for the farnesylation of proteins. Okay, just 1 second. That is kind of important. Why don't I have that slide? Okay, let me quickly. Farnesyltransferase is the enzyme that does that. So, so there's a lipid known as farnesol. It's unsaturated lipid, and many proteins are actually attached to the cell membrane using this lipid. Now let me see. Farnesyl pyrophosphate, farnesylation, prenylation. Okay, so let me leave it at that. And maybe I'll just put this on the slide. Okay, just leave it. Okay, so this lipid can be attached to proteins, and same idea as with these fatty acids here. Okay. So this one is the farnesyl, but the structure is not quite shown. But you can see basically this sulfur, if you think about, so this hydroxy group can be turned into a leaving group, right? Specifically through phosphates. We'll turn it into a good leaving group. And so, so that's one way. So it's another way proteins can be attached to cell membranes through farnesylation. And so it's intermediate in lipidation of proteins. Primary sequence of proteins, signature for farnesylation is cysteine, amino acid X can be methionine, serine, glutamate, or alanine. So, so reaction catalyzed by the enzyme known as farnesyl transferase. So there is a particular protein, RAS, which is involved in signal transduction, but it has to be attached to the cell membrane. So in other words, this one could be RAS protein, which is a protein tyrosine kinase, and it's involved in signal transduction. But the only way it will transduce the signal into the cell is when it is farnesylated. Okay, when you said that why is it on the inside and not the outside, is because that's what its function is. It receives the signal from the outside, let's say through some kind of growth factor receptor, and then starts phosphorylating tyrosine residues on other proteins. So the signal goes inside the cell undergoes cell division, and oftentimes this RAS protein is mutated, which makes it an oncoprotein. So oncoRAS compared with RAS is RAS that has been mutated and is constantly in the on state, right? So basically it doesn't need growth factor anymore. Basically all it does is just continues phosphorylating proteins and making cells undergo cell division without control. Okay, and so one strategy for cancer therapy that was investigated for many years was to inhibit this enzyme, farnesyl transferase. So now think about it. So what happens if you inhibit this enzyme? So farnesyl transferase will farnesylate RAS. So if you inhibit the enzyme, what's the outcome of that?
So it won't be able to go into the membrane?
So it will not be able to go. Is that what you said?
Yes, sir.
Yes. So if you inhibit the enzyme, you will not be able to attach the farnesyl tail to the protein, to the RAS protein. It will not be able to go to cell membrane. And then what will happen? So it can go to cell membrane. And what's the outcome of that? You break the signal transduction cascade, right?
Right.
Because it has to be there for signal transduction cascade. If you break the signal transduction cascade, you stop cell growth. Unfortunately, many of these were tested in clinical trials, but none of them came to the market. It's one of those things that, you know, the farnesylated proteins which are substrates for farnesyl transferase, they are too important. They're too important for normal cells. And so the toxic side effects were more pronounced than scientists expected. So, so you can see here that by, this is another strategy for combating cancer, is by actually breaking out the interaction of proteins with the cell membrane. Unfortunately, in this case, it was not effective. All right, so we did talk about this. Yeah, removed by detergents, span entire membrane. So these are integral membrane proteins. All right, so many of these span the membrane several times. So for example, this can be carboxyl terminus. This is the amino terminus, and this one is a 7. This spans the cell membrane 7 times. And obviously, again, these alpha helices, they're composed of hydrophobic amino acids. So transmembrane segments are predominantly hydrophobic. So tyrosine, tryptophan, these are partially hydrophobic. They cluster at non-polar polar interface. Let's see where they are. Yeah, you can see here. So this potassium channel, right? So so the silver one are hydrophobic amino acids, and the blue and the red ones cluster in the non-polar polar interface. And why is that? Because tryptophan and tyrosine, you know, tyrosine has a hydrophobic portion, but it also has a hydrophilic portion, right? Hydrophilic portion. So this is tyrosine. And obviously charged amino acids are only found in aqueous domains. So these are various transmembrane proteins. Now you can see also that alpha helices is not the only type of these. Beta sheets can also be a three-dimensional structure of proteins that cross cell membranes, right? Again, they will, and obviously they will be rich in hydrophobic amino acids. And so you can see here, all of these are hydrophobic. Now it's actually quite interesting that there is a little segment here with some hydrophilic amino acids. I don't quite know what that is about. Does it say to us? Let's see. Each red hexagon represents a tetrasaccharide containing. No. Okay, the relative size of the saccharide. So this segment here that I was just wondering about, 64 through 74, this one, right? This one has some hydrophobic residues, and probably penetrates. Yeah, does anybody understand what that means? The segment from residues 64 to 74 has some hydrophobic residues and probably penetrates the outer face of the lipid bilayer. Yeah, I don't know what this little intestine looking type structure is. I need to look into this. Maybe I'll have an answer for you on Wednesday. Okay, so where are we? We are close to be out of time. Now let's finish here actually. This one will be a good slide to finish. So there is what's known as hydropathy plots that can predict transmembrane domains. And specifically, there's a way to measure the hydrophobicity of a particular amino acids, and something which is known as a hydropathy index. And so, for example, glycophorin, which is primarily a polar protein, which most of which is exposed to the aqueous medium. You can see most of it has hydrophilic amino acids. Just one small portion, which is embedded in cell membrane, right? Bacterial rhodopsin, on the other hand, is actually a protein that crosses the membrane. And can you tell me, so it's a transmembrane protein, can you tell me how many times it crosses the cell membrane?
7.
7, that's correct. Because there are 7 humps in the hydropathy index plot, in the hydropathy plot. So if this is cell membrane, so we start on the left where it is hydrophilic, where it's hydrophilic. 1, 2, 3, 4, 5, 6, 7, like so. And so this area here, residue number, let's say we'll call this A. Where is this A? Can you find this A on this picture?
It could be the far left or the far right, where it's outside of the membrane.
Yeah. So we don't know that. So we don't know if it starts on the outside or inside. But let's just pick one. So this will be A. And then where's B?
It would be like the dip that's inside inside the cell right there.
Okay. Now there's a C right there. Now a little overdid it. So the D here, there's almost nothing here, which probably means is that it comes out and immediately goes back in, like so. So this, that little tip is not there. I hope it's not gonna erase the whole thing. Yes, it did. Anyway. So, but you got the idea, right? You got the idea. Like that. And then so if this is D, E, so E will be here. D will be here. So here it doesn't come out so much because there's almost no hydrophilic amino acids. Here it comes out quite a bit. EF. And then we have, it looks like it doesn't come out here at all, right? So G will be something like that. All right, this makes sense? All right, so these are hydropathy plots, and you can see you can use them to predict the transmembrane domains, just based on the hydrophobic, hydrophilic nature of the amino acids. All right, guys, do you have any questions about today's lecture? No? So look at that paper again, and obviously more about membranes, and there'll be lots more about membranes on Wednesday. All right, the homework is open, and let me know if you have any questions.
Thank you.
Yeah, bye guys.
Thanks. Bye.